In this video, we'll discuss the FDA regulatory guidances for antivirals discovery. First, you need to search for the FDA guidance documents. This is very easy. When going to the FDA website, search for guidance document search. You will find a very big table and you will appreciate that at the moment there are almost 3,000 entries. You can either type a search term, for instance antiviral or virus, or also search by topic. Most of the guidance documents are under clinical and then medical. FDA guidance documents are not binding, neither for you nor for the FDA, meaning you can choose or the FDA can choose to apply them or not. However, you'd certainly be advised to know them, as the FDA will often refer them to you. Most guidance documents are on general processes for discovery and development. I will not discuss these today, as there are a number of very good YouTube channels on the subject. I present here one pharma guideline. Instead, I'll discuss today only the FDA guidance documents applicable to all antivirals discovery and development. There are 4 of them. The first one on conducting and submitting virology studies, which dates from June 2006, so already a while ago, and its associated guidance documents specifically for HIV, HPV, HCV, and influenza, as well as their revisions. You can find the link here below. The second document is on submitting next-generation sequencing data for resistance mutation studies, both preclinical and clinical. This one is obviously more recent, as the technology itself is also more recent, so from July 2019. The third one is on clinical development and labeling of antivirals. This is a very old guidance document from February 1997. And then the last one and fourth one on the development of anti-infective products in pediatric populations. This one is a more recent one from December 2021. I will mainly focus on the first 2, as they are also applicable to preclinical data, and actually mainly on the first one. The second one on NGS data contains only very obvious data that you would like to share with the FDA when you submit such information. So let's have a look at the first guidance document on conducting virology studies for antivirals discovery. So let's assume you've identified the perfect antiviral candidate and you want to push it into clinical development in the USA. The first set of recommendations from the FDA is to clearly identify its mechanism of action. For this, you can either use a replicon assay if it inhibits a viral polymerase, or an enzymatic assay / binding assay, depending if it inhibits a viral enzyme or a viral protein. You will also need to demonstrate specificity over host enzymes / homologous proteins to demonstrate it only targets the viral protein and not any host target. And you can also demonstrate MOA by localization of resistance mutations. Here, a clear comment is to sequence all viral proteins which are part of functional complexes, as sometimes resistance mutations can be found a bit further away than the actual target. If your antiviral of interest is an immunomodulator, meaning it does not directly target the virus but rather boosts or inhibits the immune system, then you need to define the precise components of the immune pathway which are targeted by the compound. You also need to precisely demonstrate what the binding site of your product is. This can either be done by structural biology, X-ray or cryo-EM, or by studies such as HDX or cross-linking. Here, the advice is to not directly use resistance mutations, as they can sometimes occur far away from the binding site. A next set of FDA recommendations is on in vitro antiviral activity. Here, make sure to use concentrations that can be achieved in vivo. in the tissues of interest. So not only in blood, but in the actual target tissues if the virus replicates there. So you need to have an idea of the PK of the candidate. Make sure to use low passage numbers for cell lines as well as primary human cells if possible. The FDA will request you to check antiviral activity on multiple viral strains and clinical isolates, making sure that they are currently circulating, that they have a wide geographical variation, as well as variation in genotypes and phenotypes. As much as possible, include strains resistance against medicines which are already currently on the market. A next recommendation, which I actually haven't seen applied too often, and I think is a very good idea, is to check the effect of increasing MOI on the antiviral activity. You may also want to include time of addition studies, although in my experience, they are not always required. Finally, if you observe a discrepancy between the potency of the candidate between cell-based assays and biochemical assays, it will be requested to understand why. This is often observed for nucleoside and nucleotide analogs. And there, the advice from the FDA is to measure the intracellular concentration in triphosphate form as well as its half-life. For small molecules, the FDA advises to evaluate the percentage plasma protein binding, and if it is high, to measure the antiviral activity either in presence of human serum at different concentrations, so 5, 10, 20, and 40%, or in presence of physiological concentrations of human serum albumin and alpha acidic glycoprotein. From these, you should derive a serum adjusted EC50. While this is the FDA recommendation, I do think there is a better approach, and it is to rely on free concentrations, both in the in vitro assay as well as in the target tissue, and you can correlate both. Also remember that even if plasma protein binding is high, it is not necessarily an issue if the affinity or the K-off of the antiviral compound for the viral target is very high. Indeed, even if there is a lot of plasma protein binding, if the affinity is very high, all the viral targets will be saturated even with a very low free fraction. Remember that you're dealing with an equilibrium between viral target, compound, and plasma protein. After measuring in vitro antiviral activity, it is definitely essential to look at in vitro cytotoxicity. Here, the FDA recommendation is to not only measure cytotoxicity in the cells that were used to establish the antiviral activity, but also in multiple different dividing and non-dividing cells. For nukes and non-nucleoside analogs, make sure to evaluate the bone marrow toxicity potential in human bone marrow progenitor cell colony formation assays, as well as measure the inhibition of human DNA/RNA polymerases. Finally, the FDA advises to look at mitochondrial cytotoxicity by measuring either the mitochondrial DNA content, the morphology of mitochondria, lactic acid production, glugal assay, seahorse, etc. Next, the FDA has a number of recommendations which make a lot of sense for in vivo antiviral activity. The first one is to not only measure viral load, record also all the other parameters which may be useful. So mortality, clinical observations/morbidity, histology and histopathology, and pharmacokinetics, both in blood and in target tissues. This one may often be forgotten in some academic settings. The last recommendation I have not always seen being performed and is actually a very interesting one, and it is to isolate and sequence the potentially resistant viruses generated upon viral rebound. It's a kind of in vivo resistant selection experiment. So in a way, it could be even closer to what would be happening in humans. Next, the FDA has a set of recommendations for in vitro resistance selections. The first one is to establish the multiple potential resistance path. So make sure to include repeats in your experiment. And also make sure to include different product concentrations. Indeed, you may more easily observe mutations at low concentrations of product. You can either do this by increasing concentrations during the assay or performing the assay with multiple different fixed concentrations. For each separate substitution, as well as combination of substitutions, make sure to evaluate the potency of the product, but also of the marketed competitors. You will obviously perform the first one, but sometimes I've seen the second one being forgotten. Also make sure to evaluate the fitness of the mutant viruses. Compare your results to products of the same class. On the other hand, make sure to evaluate the potency of your product on strains which are resistant to marketed competitors, as cross-resistant is not always reciprocal. Finally, if you're developing a combination of products, make sure to evaluate whether these are truly synergistic or additive. And even if you're developing a single product, make sure to do checkerboard experiments with the marketed products. Indeed, it is possible in real life that your product will be used with an already marketed product. You need to make sure that no antagonism will be observed in that case. All right, that was it for the general guidelines for all antivirals discovery. Now moving to the virus-specific guidance documents. As you can appreciate below, there are a number of them. The idea is not to go through each separately, but more to list them for your own information, so you can go and check them. Most of them are on HIV, as historically a lot of HIV antivirals were discovered and developed, as well as HCV, HBV, HDV, influenza, RSV, rabies, smallpox, CMV, HSV1, SARS-CoV-2, and more recently Mpox. For some of the older guidance documents, there are also revisions in the meantime. So make sure to check whether your virus of interest is included in a guidance document. Finally, I want to let you know that the FDA itself has a YouTube channel. On it, you can find some very useful playlists. The first one I want to highlight is the CIDR Small Business and Industry Assistance, which is edited with new videos and a new playlist every year. So here you can find the link and you can see that there is one for 2025 as well as 2024, etc. And I think they started since 2017. I also highlight two other interesting playlists. The first one on data requirements for biosimilars and the second one on biomarker development. Now up to you. Do you have any additional advice on FDA guidance documents for antivirals discovery and development? If that's the case, do leave a comment